Sayed from Drbeen.com. Welcome to one more show. So the discussion today is going to be brief but interesting, and that is what is ApoB or apolipoprotein B. There are many kinds of apolipoproteins, but ApoB is important for us to discuss today because I want to start a series on the cholesterols and LDLs and triglycerides and the role of ApoBs and the new studies that Nick and his team are doing, Dave Feldman's work, the previous traditional work, and the how LDL level and cholesterol levels are used, and then how ApoB may be a better measure, and then maybe not in certain cases. So I want to address those things and talk about them. And it is important for us to have these foundations available. So today's discussion is going to be ApoB. What is this molecule? So let's start with the references first. So this is Drbeen.com. And if you would like to have access to another tons of lectures, just go to Dr. Bean and see the plans there. I had actually another idea as well that I wanted to ask you. For general public who doesn't want to have medical education in general, would it be interesting, because we have finished the lifetime, would it be interesting to have you access to the lectures for $3.99 a month? So it's not lifetime, it is recurring, but it is a lower price. I am doing some work that is only going on Dr. Bean and not on YouTube. That may be interesting. Plus, I do some webinars on Dr. Bean as well, which do not go on YouTube. So let me know if that is something that you would be interested in. If so, then I would get those plans done and share them on YouTube. Okay, so this is Dr. Bean. This is one, biochemistry, apolipoprotein B, and all about it. These are all the links that are present. Then this is apolipoprotein B, ApoB-100. So this is an enzyme that we would discuss today. Then some more references about apolipoprotein Bs and their functions, etc. And as you can see, there are a lot of references here. So with my drawings, apolipoprotein, although from 2010 onwards, there has been a lot of interest in apolipoprotein B and its atherogenic role, or the role in blood vascular damage, especially the blood vessels of the heart and the brain, resulting in cardiac events, coronary events, and strokes. The interest peaked in 2010 and it is continuing. But please remember, even in 1970s time, there were studies discussing apolipoproteins and their atherogenic role, especially these proteins. So it's not new, but there is more evidence. At the same time, the evidence for these proteins to be related to cardiac risk and stroke risk or coronary event risk is still developing. You would see there are a number of folks that believe strongly that this is the only and the most important measure to have to understand the risk of coronary or stroke events. There are some others who do not believe that. There are yet others who believe that traditional LDL and cholesterol measures are the measures. Then, of course, I'm sure that you are aware that we had Dave Feldman with us. So Nick Norowitz and Dave Feldman and their work that shows very high LDL and VLDL levels, which also means very high ApoBs levels. And then they're doing research on the outcomes, and it seems that their initial studies don't show the damage. And so we would follow that with interest. My point is, it is an evolving science. And for us to understand the foundations is easy because then you can think in whichever direction you want. Okay, so the story of ApoBs. ApoBs, apolipoprotein B, has two isoforms that are interesting for us. We are talking about B. There is actually apolipoprotein A and apolipoprotein E and so on. ApoB has two isoforms, ApoB-48 and ApoB-100. That is what we are interested in today. So these two isoforms, ApoB-48 and 100, the 48 is made primarily by the intestinal cells. So when you eat something that has fats, and our body wants to package these fats into little packets that would be able to swim in the blood circulation and tissues, interstitial fluids. These are apolipoprotein particles or lipoprotein particles, as they used to be called before. To package them, to package fats in those particles, the intestinal cells make a structural protein called apolipoprotein B or ApoB-48. And we'll come to the number that why is it called 48 in a second. So the intestinal cells make that protein, then they hang triglyceride and cholesterol esters and create a phospholipid layer around all of those packets and make that little chylomicron and send them off into the blood circulation. We will discuss about them in detail and their structure and their function and their benefit or their damages in the future talks. Today, just this much, that the structural part of these particles are apoproteins, especially here ApoB from intestine to package and carry the fats in chylomicrons. Then our liver cells or hepatic cells, they also manufacture apoproteins, ApoB protein here. In this case, that is what we're talking about. Liver makes ApoB-100. And why does it make ApoB-100? We'll talk about that. What is so special about the liver cell that it makes 100 and the intestinal cell makes 48? We'll talk about that too. But just remember, for the time being, these two are important ones. When liver makes ApoB-100, it tags that or it uses that as a structure to, for example, make VLDLs and LDLs. So these VLDL, IDL, LDL, these particles have this backbone on them or the structural protein on them, which is ApoB-100. Now, how is the ApoB-48 formed or made, and how is the ApoB-100 made? So the first thing to keep in mind, the number 100 or 48 actually talks about the percentage. ApoB-100 means ApoB 100%. ApoB-48 means ApoB 48%. So what does that percentage mean? So let's look at that together. So ApoBs have their genes, ApoB-48 and 100. They both have one gene actually, or one genetic area, that is on chromosome number 2, petite arm, small arm, location number 24. So in this location, there is a gene that when transcribed, that means when you, so imagine that this is a nucleus, and from this nucleus, when we transcribe this gene and we make messenger RNA, which then gets translated with ribosome. I've done this discussion before. In general here, the messenger RNA comes out. Messenger RNA is then read by ribosomes. Ribosome read the instructions on the messenger RNA and they make proteins from there. Right? So this is called the process of translation. Every messenger RNA has a stop codon in it. Stop codon tells the ribosome where to stop, where to disengage from this messenger RNA. So this ribosome will start reading from here and translating that into this protein, and then will continue going until the stop codon is found. Good? So when this messenger RNA reads up till this stop codon, the original stop codon, then the protein that is formed is called ApoB-100. This is 100% of the protein that is formed from the reading of 100% of this messenger RNA. There is an important thing to notice, and that is on this side of the diagram. Notice this, I made this little claw or hand here. When you translate the whole protein, a whole RNA into a whole protein, the last part, the carboxy part of the original 100% of the protein is actually a receptor called LDL receptor. This is the receptor that is on, so if I go here, this is the receptor, let's say here, that is on these particles, for example, VLDL, LDL, IDL, especially LDL, which allows this particle to bind with the receptors, with their ligands. So that is an interesting thing to keep in mind, that there is a receptor ApoB-100. Now, ApoB-48, remember ApoB-100 was made in the liver, correct? And it was tagged on VLDL, IDL, LDL. And it has a receptor on it which allows it to go and bind with various ligands that it has on the tissue to give cholesterol and do its function. Now, if I flip through these two couple of times, you can see the difference between them. In the intestinal cells, we make ApoB-48, as I said before. This ApoB-48 gets connected to chylomicrons, as I said before. Now, the gene is the same, chromosome number 2, petite arm, the small arm, and the location number 24 has the gene for ApoB. I won't say it is for 48 or for 100, it is for both. But in the intestinal cells, when this messenger RNA comes out in the cytoplasm, there is another group of enzymes that are present in the intestinal cells. These enzymes quickly rush to further modify this RNA. They are RNA editors. They're not present in the liver, or even if they're present, they're not active. They're present in the intestinal cells. They become active. And what they do is, it's such a beautiful thing. At the location, messenger RNA base number 6666, no, I'm not kidding, this is the number, 6666, at that point, what they do is they quickly go and make one base change. That change converts this messenger RNA's area here into a stop codon. So when that becomes a stop codon, now we have the translation by the ribosome that would start from here and go up till here, because we have a stop codon here. And it would ignore all this instruction set. That means the protein that it would make will not be all the way to that LDL receptor. We would end up with 48% of the protein. This is why it is called ApoB-48. It has 2,512 amino acids, while ApoB-100 has 4,536 amino acids. So let's just very quickly look at this reference. I really love this enzyme. This enzyme is this one, ApoBEC1, Apolipoprotein B, mRNA editing enzyme, catalytic polypeptide 1. It is also called C to U editing enzyme, cytosine to uracil editing enzyme. And if I go in here, look at this. By converting cytosine to uracil, A1 changes the codon from CAA to UAA. Did I write it correctly over here? So there was CAA originally, and then they convert that to UAA, which becomes a stop codon, and the translation only goes on till 48%. So that is the protein that we get. So of course, this protein doesn't have LDL receptor at the end. This is why the chylomicron has the ApoB-48 on it, but it doesn't have that capability of binding with the LDL ligands like ApoB-100 has. What a beautiful mechanism. Okay, now one more thing. Why does this enzyme do it in the intestine and not in the liver? Why does liver makes 100% of the protein and intestine makes 48%. We know that the functional outcome, from the functional outcome, we know the benefit of it. But why do these cells coordinate themselves in this way is a very interesting research area as well. And of course, that is also interesting research area to see if we can make medicines that would then cause this LDL receptor not to be formed. And there have been some medicines that have been made. The problem so far has been that they are useful. They reduce the LDL's capability to bind. They reduce the ApoB's production in the liver, ApoB-100's production, etc. But there has been liver side effects because of those. Those medicines aren't yet fully out. So, very interesting topics. Now, just another quick reminder that what is the role of ApoB? Why are we now focusing on this protein? To say that this protein can also cause atherogenesis. It can also cause or contribute towards the plaque formation. Why? So here, normally, imagine that the blood vessel internal cells, the endothelial cells, the inner surface of the blood vessel, imagine that surface is sort of a swimming pool for these lipid particles. And they dive into it and they go under it and then they come back out. So this is their normal behavior. They're like little kids who are diving into a swimming pool and then jumping out of it. And that's fine. They go into subintimal space. Intima of the blood vessel, especially coronary blood vessel, is that we have these endothelial cells or the inner surface. Below that is an elastic area with these elastic fibers, because remember, the blood vessels of the heart not only have to dilate in diameter, they have to, with the heart, they have to increase and decrease in length as well. And we don't want that every time they reduce in length, they become kinky or they develop a kink in them. And when they are stretched, they become so thin that they are stretched thin. We don't want that. So these blood vessels are very much designed in a way that they can easily, elastically become lengthened and shortened. So for that, there is this special elastic fibers and connective tissue right under the endothelium. Then below that, there are smooth muscles of the blood vessel wall. And between the smooth muscles, which is the tunica media, or the middle layer, between the smooth muscle and the intima, which is these two layers, is a space. This is called subintimal space. And these little particles, lipid particles, they dive in, they go in here, they come out. And that's the normal behavior. However, in some cases, that normal behavior becomes slightly disrupted. What is the disruption? The disruption is here, if the number of lipid particles, these apolipoproteins, for example, let's say LDLs, if the number, that is concentration of these particles, is high, once again, keep in mind Dave Feldman's work and the Nick Norowitz's work, where they are looking at very high concentration of LDLs without effect, or at least I understand without effects. Maybe there are effects, they'll find more. So it's not, I cannot make a generic statement that when the LDL is high, it's always going to cause a damage. There are so many studies that show the damage. There are now newer studies that show in some people a specific phenotype, this may not be happening. So we'll see. But generally, as there are more concentration, as there is more concentration of these lipid particles and they dive in, they start becoming captured in there just because of their number is so much that they just go in and just keep getting trapped and getting captured. So that is one possibility. The second possibility is that it is also possible that if somebody has taken a lot of fats, or they have some genetic issues, whatever it is, let's say the lipid particle size is bigger. And that big blobby particle gets in and releases some cholesterol, plus it is big enough that it gets captured in there. That is also possible. And I will discuss that a little more in a second. Another possibility is that the tissue is inflamed. The blood vessels are inflamed. The body is inflamed. There are lots of inflammatory mediators running around. Let's say nowadays COVID. Let's say long COVID. Inflammatory mediators. Let's say chronic inflammatory states. Any chronic inflammatory state would have more inflammatory mediators present in our body. When these are present in our body, that those inflammatory species, like reactive oxygen species, can cause damage to these particles and the cholesterol in them, resulting in inflammation. So I'm going to talk about that in a second. From this diagram, just this much, that the concentration, if ApoB's concentration is more, that means the number of particles that have ApoB, number of particles are more. Or if the sizes are more, that means a lot of cholesterol. They both can be atherogenic. So it is not true to say, well, only the cholesterol will be atherogenic or only the ApoB will be atherogenic. They both can be, and I'm going to explain. So here, let's say that this is an ApoB particle. Let's say this is an LDL particle. And let's say it has more cholesterol than normal. What that will mean is that when it comes into this subintimal space, it will spill the cholesterol. Imagine this person has no inflammatory mediators, no inflammation, all clear. Although we all have some inflammatory mediators, but let's just give it no inflammation. There is this particle. The particle is going to give the cholesterol out. When that cholesterol is squeezed out of this particle, the local inflammatory cells will eat up that cholesterol. They will become foam cells. They'll become little fat obese cells with the cholesterol in them, which would cause them to start releasing inflammatory mediators, and inflammation will occur. Let's say there was no inflammation, but now inflammation has started. And then more inflammatory cells will be, or more immune cells will be brought in there, and more inflammation would occur, which would cause the local smooth muscles to react, and they would increase in number. They would try to kind of cover this thing and trap this thing. So there will be a tiny, thin roof that would form on plaque, which could then open up and cause problems. So cholesterol itself, when leaked in these vessels, can trigger inflammatory process. On the other hand, if the ApoB itself is there, which, as I said before, ApoB particles can dive in and dive out, but sometimes they can get trapped. What happens when there is inflammatory state? When there is high oxygen, reactive oxygen species levels present. How could these be present? There are so many reasons. For example, light or radiations can do it, certain chemicals can do, smoking can do that, certain foods can do it, certain toxins can cause inflammation. So there are so many reasons for chronic inflammations or inflammatory mediators. When they are present, look at this one. Let's say this is a reactive oxygen species. Reactive oxygen species can directly attack the protein ApoB and deshape it. Now, this is a very delicate point, and please keep in mind, it can deshape it. It can change the shape of the protein where it is attacking. Do you know why it is a delicate and important point? Because where it is attacking, imagine somebody is carrying a hammer and they're hammering away on a wall. When they will get off of it, you would see that the wall has a new pattern on it. It's a broken tissue structure or wall structure. Similarly, when reactive oxygen species would hammer away or attack the protein, they would create new epitopes. They would create new shapes there. Our immune system is not used to seeing those shapes. So what would happen is immune system would start attacking these ApoBs, and we would have an immune reaction. Similarly, we can have lipid peroxidation. Remember, this whole particle is filled with lipids, right? So reactive oxygen species can attack lipids. Lipids in turn can become bound. They can stick to the ApoB proteins. Now, lipid peroxidation occurred, that is, reactive oxygen species attacked the lipids. Now, lipids have become reactive. Now, these lipids are binding to ApoB. That ApoB, which is now bound with the peroxidized lipids, is atherogenic. That means it would cause inflammatory reaction. So, reactive oxygen species directly attacking the ApoB will make it atherogenic. Reactive oxygen species attacking the lipids then in turn becoming peroxidized, which then bind with the apoproteins, causing them to become atherogenic. So imagine it this way, that if we have ApoBs, which we all have, and imagine that these are like a structure that can catch fire, but there is no matchstick to burn them. And now imagine that there is inflammation, the inflammatory state in the body is high, which is equal to little dynamite sticks or little matchsticks. So as these matchsticks kind of cause ApoBs to become D-shaped, inflammation would start. So inflammatory state plus ApoB is more dangerous than just the ApoB. So I hope this is clear. Now, finally, what happens is, for example, here is cholesterol. It is also possible that the cholesterol can spill, and it happens. There is, of course, huge number of studies that show this. When the cholesterol still within this subintimal space of the blood vessel, when the cholesterol spills there, that would cause the local inflammatory cells, macrophages, to start eating it up and become big foam cells, and then those foam cells can cause inflammatory reaction and plaque formation. So I wanted to read this quick thing and then we are done. Here, oxidative modification of LDL lipids, oxidative by reactive oxygen species, modification of LDL lipids and ApoB-100. So let's say this is the ApoB-100, and then there are lipids in here. When oxygen attacks them, that's what they're talking about. Remember, ApoB-100 produced by liver attached to, or then produce the whole LDL around that. By reactive oxygen species is widely regarded as a crucial event in atherogenesis. ApoB-100 modification, for example, binding of lipid peroxidation products, lipid peroxidation product, or direct oxidation of amino acid side chains, direct oxidation of the amino acids, residues, are thought to finally result in the formation of a new epitope. It creates new shapes here that are specifically recognized by scavenger receptors. Scavenger receptors are the receptors. Let's say this is a macrophage. These receptors here, they're scavenger receptors, like toll-like receptors. They're sensing, they're trying to figure out what's going on in the environment. Is there something damaging in the environment? These are scavenger receptors. So they are sensed by the scavenger receptors. However, despite convincing evidence from in vitro and animal experiments, data concerning the role of oxidized ApoB-100 in the development of atherosclerosis in humans in vivo are scarce. One reason for this is the shortage of sensitive and specific methods for direct measurement of oxidation in circulating LDL. And this is an area where the science would continue to evolve. So whatever is the current state, we will actually go beyond that. And the interesting thing, why it is said that ApoB-100 may be a better measurement of identifying the risk of coronary artery disease and coronary events and strokes, is that one ApoB molecule is on one lipid particle. So if you wanted to measure exactly the concentration of the particles, which matters, as I discussed, not just the cholesterol within the particle, but the concentration of the particle means more ApoBs. That would mean more chances of atherogenesis. Then measuring the ApoB seems to be a superior metric over measuring LDL or cholesterol levels. But at this time, traditional measurements are still with the LDL cholesterols, and that is fine. That is what's happening. ApoB is slowly coming into the mix as well. And what would happen next? what new phenotypes, where would it be more useful, where would it be less useful, we'll find out. But this is the discussion of ApoB. This is the story of ApoB. We will now continue in this series with other discussions, and we'll go from there. Thank you very much. Please like, subscribe, and share. Tell me again if you would like to have a plan for $3.99 per month to have all of these health, aging, and nutrition related topics on Dr. Bean. Thank you very much. I would see you next time. Have a nice weekend.